Yeah, thank you all so much for having me. It is such a delight. I'm part of the DARE HIV Collaboratory, and I was talking to a bunch of other folks who work on HIV research, and I was saying how much I hope that I get an opportunity to do more work with our CAB in DARE, and then also got this invitation at the same time. And so I'm really pleased to be here. I don't have too many slides prepared because I know everybody is very zoomed out, and I also thought it would be really nice to just kind of hear, to start with kind of hearing all of your perspectives, or some of your perspectives just about what's on your mind about this topic of kind of like, how do we think about mRNA vaccination for COVID, which has obviously been a big thing, and how that informs HIV vaccine design. Before I go into sort of talking about some of my thoughts. But as Lauren said, I'm a doctor and I'm a researcher. I have a lab based at San Francisco General Hospital, and I mostly study the killer CD8 T cells, which are not the main ones induced by most vaccines. But we think a lot about sort of how do you form a good immune response against infections, and the work that we're doing in HIV is mostly in the kind of HIV cure context of trying to figure out, like, can we get these cells to suppress HIV as kind of like a cure modality. And my clinical work right now is I take care of patients at Ward 86 at the Ward 86 clinic. I came out to UCSF for my residency and stayed for my infectious disease fellowship, and so I've been at Ward 86 since 2016, and that, I would say, my patients are what really drive my research in terms of sort of thinking about why we're doing what we're doing. And so I'm super excited to be able to sort of talk. I get really dorky sometimes, so you can definitely stop me if I get too dorky, but I'm really excited to talk about vaccines and immunology. And so maybe we could sort of start with that question of just can you tell me some of the things that are on your mind about sort of the connection between thinking about the COVID vaccines, the mRNA vaccines, and HIV vaccine development. And I'll go through my slides, and there's some other opportunities for like sort of discussion and reflection, just thinking about things.
So I guess I have a question. I'm not a CAB member, so I'm actually in research partnership management position, and I help coordinate the CAB. But I'm curious if the HIV vaccine is also an mRNA vaccine. I guess COVID was the first time that I sort of I've heard about mRNA before, but as a vaccine. So anyway, I'm curious.
That's great. And I have some slides because I kind of figured like the mRNA vaccines, I mean, I guess now we like it's like headlines in the news, right? But I have a couple slides sort of going over a little bit about the history, because I feel like that gets a little bit lost in the, I don't know, the pressure of the pandemic as it's been. So that's a great question. What else is on people's minds as you think about kind of this connection between the vaccine, these latest vaccines, and sort of thinking about what they might be able to do for HIV?
Well, this is Jesse. I'm a CAB member. And my question, I guess, is the obvious. If they could do the COVID vaccine, why didn't they do the HIV vaccine?
Yeah. Yep. That is also a wonderful question. And so this is good. I'm glad. I was trying to figure out, I wasn't exactly sure who was going to be here, what people's questions were. And so I have a few slides sort of addressing some of the unique challenges of thinking about designing a vaccine for HIV, and would very much encourage people to interrupt me as you have specific questions, or just make sure I don't get too dorky on the immunology side of it. Other thoughts people have had in terms of thinking about this question? Dork away. Oh boy. I probably will, regardless. Do people feel like
only
sorry,
my only other thought, I mean, I think those were great questions. I mean, just in terms of, there's been a lot about like memory cells and I mean, I know this is more about COVID vaccines, but I guess since you're here, it would be, the fact that if people's antibody levels are falling after a certain amount of time, but they still have the memory cells to respond, to create a quick response. And so anything about like sort of just how the vaccine mechanism works for long-term immunity once people's immediate antibody response goes away, and you know what I mean? Something like around that.
Yeah. And that's funny. I study memory T cells. I did not put a specific slide in about sort of memory responses, but there'll be a spot, I think, where it will make sense to talk a little bit about, what does the immune response look like after a vaccine? Does everyone feel like they've learned a little bit more virology and immunology over the last, couple years from the pandemic? I know I get a lot more questions of types of questions from my family that I never got before about memory T cells and antibody responses and neutralizing antibodies and things like that. It's very interesting, certainly, to see all those discussions, and I'm certainly happy to sort of, I obviously love talking about immunology and virology, and so it's nice to kind of have those discussions and sort of think about it together. Carolyn, I think I see your hand up.
Yeah, my question is more on the controversial issues of vaccines right now, which is a real concern of mine. We're very divided as a people right now in how people feel about vaccines. So I was wondering if you could talk a little bit about what is the scientific community doing to bridge that gap, to heal those wounds of distrust with science, with the science community, and also with the pharmaceutical community, because the reality is this pharmaceutical community or the pharmaceutical companies, I should say, I don't even think of them as communities, but they end up being the ones that develop these vaccines. And so what is the role of the scientific community to help with some of those issues that we're having right now in trust, with the public trust when it comes to what's going on with the developments of vaccines right now?
Yeah, I think that's great. I'm actually, like I said, I don't have that many slides. I'm putting a couple of these questions at the end so I can remember to address them. I think that's something I really take to heart, and I think something that only comes through kind of natural, organic conversations and community building between pharmaceutical companies and communities in order to understand what the different kind of perceptions are. And I think no one knows that better than people in the HIV community, right? In terms of the history of sort of building those connections and pushing the scientific community and pushing pharmaceutical companies and industry to think along the lines of what the priorities are for the people. I think that's a really important conversation, and I put a note to myself to come back to that at the end, because I maybe as we talk about some of the specific immunology and sort of the specific development of these vaccines and the history of them, I think some of that informs a little bit in terms of thinking about, what is it that can be, what are the things that can be better communicated to help encourage, build trust in sort of how the science is being pushed, I guess. And Jesse, I see your hand up too.
Yes, hi.
Hi.
This is very interesting. So the question I have too is, when the vaccine first came out, they talked a lot about immune suppressed communities. And for me, being HIV positive, I felt like there was really no direction from the HIV community to address HIV positive people, to let them know that we're in that class because we are undetectable. I mean, because that we're not in that class because we take our meds adherently and we're undetectable. So who were they talking about in HIV?
in terms of priorities for getting vaccinated and priorities for getting boosted. Yeah.
Yeah. And just throwing that word around, period, immune suppressed. I mean, every time I hear it, I think of myself, but I'm really not, because I'm adherent and undetectable.
Yeah, I've had this exact conversation with, I feel like, almost every one of my patients over the last few months, as we've come up with this question about boosters. I think my perception is that this question is very much kind of contained within, it's been very confusing to figure out who should get a booster, both from sort of a scientific standpoint and then also communicating that at kind of the CDC level and public health level, because there are a lot of nuances. But I don't know that I'm the best person to answer the question about sort of how that communication happens. I haven't, it's a really good question that I haven't sort of thought about, sort of how the, how to think about this exactly. And maybe that's a discussion we can come back to at the end too, about sort of the general question of like, how do you take all this science and this immunology and these exciting developments and actually, I guess sort of help bring it to people in a way that that they can that people can understand and that people feel like they can have trust and confidence in. And that seems to me sort of like a part of that discussion too. Okay, Jesse, I can't tell if your hand is still up or if the Zoom thing is always so funny, or if that was from, okay.
It's down now.
Okay. Great, well, like I said, I don't have too many slides. I think I have a kind of a good sense of some of the things that are on people's minds. Is it okay if I start to go through them? Okay. Please interrupt me. I have trouble tracking the chat sometimes when I am presenting. So if someone is able to just keep an eye on that and
I can do that
tell me,
for you.
that would be great. Thank you, Lauren, so much. Let's see. Is this showing the correct screen? Okay. So this is mostly about vaccination and then sort of talking about some of the science and talking about sort of the clinical aspects of thinking about how do we think about all these developments that have kind of come to us in the last year and a half in terms of the mRNA vaccines for SARS-CoV-2 and the relationship to what we can understand for vaccination for HIV. I mentioned at the beginning, and I thought I would just do like a quick introduction of myself and my background. And so, as I mentioned, I'm a physician scientist. I do all this very dorky lab research. And I also am a clinician as well. I take care of patients. And I did my PhD really like in the deep part of trying to think about how do we form good T cell responses to viral infection in cute little mice. And so that was like my PhD background. And I like to joke that T cells are my science love. My wife knows this, so she knows that that's like a separate love, but T cells are definitely sort of my science love. And when I kind of came into fellowship and thought about what I wanted to do kind of for when I grew up for the rest of my life, I really wanted to take all these things that we know about how to make good antiviral responses in mice and apply it to people, both at the basic biology level and then also in terms of clinical translation. So as I mentioned at the beginning, most of what I do in my day-to-day research is like look at T cells, think about how to make them better T cells to fight HIV. And then also sort of look at T cells that are in people who are enrolling in our clinical trials that include kind of therapeutic vaccination, which is different from most of what I'll talk about today, but therapeutic vaccination for HIV and then other T cell based therapies, like we have a CAR T cell clinical trial that is opening soon. I think we're starting to screen people already for it. but that's really where I live now, is sort of on this spectrum of thinking about how to make effective T cells for HIV cure. And we think about T cells in the context of HIV, and we also think about it, try to learn from acute infections, and kind of another setting where T cell responses aren't always so good, which is at the beginning of human life. And we apply lots of fun, interesting, like sort of high dimensional, interesting immunology, assays to be able to really try to get at some of these questions. And so that's just kind of my background and orientation to thinking about this. When I went to Croy this year, Marina Kasky had a really nice slide, and lots of people have sort of presented these. And so I've adapted this slide. I think it's helpful for thinking about the broader ways that in kind of the scientific community and in the kind of clinical translational world, there's been so much crosstalk between work that's been done in the context of HIV and work that has been done in the context of SARS-CoV-2 infection. And I think, I'm so like grateful to work in the field of HIV, because I feel like we all felt like we could really do something to sort of contribute to understanding what's happening with SARS-CoV-2 infection at various different levels. So, a lot of the work and tools developed to look at viruses and immune responses, and then the clinical, antibodies, making antibodies for therapeutic use and having infrastructure to do big vaccine trials comes so much from the work that's been done in the HIV field. And then similarly, I think over the last coming up on 2 years, we have had just an incredible amount of better understanding of a lot of things because of work that's been done in the context of SARS-CoV-2. So both, kind of seeing these vaccine trials really advance quickly. I think there's always this tension in the clinical world of, do you want things to move quickly, or do you want to give them, how much time do you give them? And there's so much that goes into those decisions. And I think we had a situation where we needed vaccines quickly, and we saw that we were able to do that. And so that is really informative for thinking about, what are the holdups for getting vaccines approved in HIV? Are there ways to make some of that timeline shorter? And again, that's not really the work that I do, but I think we all have come to appreciate that some things probably could be moving faster. On the biology side too, there's faster development and deployment of kind of, again, mostly antibody therapeutics with the monoclonal antibodies that people have gotten for COVID. And then, I think about it from the immunology standpoint, that we have so many cohorts, and we're doing some work at the general with a large kind of longitudinal cohort, very much like the SCOPE cohort. We work with with participants in the SCOPE cohort to do our HIV studies, and we have a large SARS-CoV-2 cohort called LINK, and so we've come to understand, and many investigators have come to understand infections in humans much better as a result of kind of all of the studies that have been done in the context of SARS-CoV-2. So I bring this up because I think the question here is really interesting. Like, how do we think about mRNA vaccination and its impact on HIV? But I would say it's really, again, it's like part of this larger ecosystem of crosstalk between the two. And so this is kind of the question that we're looking at and thinking about. And I thought I would just review really quickly that, prior to 2 years ago, actually, maybe even prior to just this year, many of us didn't hear or know about mRNA vaccination, and even me, an immunologist, didn't think too deeply about exactly all of the different vaccine platforms that are sort of out there. But we get a lot of different vaccines in our lives, and the kind of classical vaccine platforms are kind of giving people bits of the proteins from the viruses. So like the flu vaccine and the shingles vaccine, the new shingles vaccine, are formulated to be like bits and pieces of the virus that is given to the immune system to educate it to make an immune response. There have also kind of been, for a long time, live attenuated viruses, right? Because the virus elicits a good immune response. So why not give the virus, just attenuate it so it doesn't cause severe disease. And that's obviously can't be given to everybody, but the MMR vaccine, the measles, mumps, rubella vaccine we get, when we're born for the most part, that's an example of a live attenuated vaccine. And then there are like these things called virus-like particles. So they trick the immune system. They look like a virus, but they're not actually a virus. And so that's the example of that would be one of the HPV vaccines. And you can also have whole inactivated virus itself. So these are, exactly. These ones that look like viruses here. This is an actual virus. This is an inactivated virus, so it can't really do anything in the body, but bits and pieces of it are used to stimulate the immune system. And this is not a virus, but it's built to look like a virus. And so those are classical vaccine platforms. And then the newer platforms that have come up in, I think to our attention in the context of the pandemic, but have been around for some time, include viral vectors. So not the attenuated, for instance, measles virus, but taking another virus. And in this case, in the case of the Johnson & Johnson and AstraZeneca vaccines, they use these adenoviral vectors. They can't replicate in the body, but they look a little bit like they look like a virus. They are a virus. They can give the signals to the immune system to make a good response. We also obviously, and kind of topic of today is thinking about the mRNA vaccines, like the Pfizer vaccine, the Moderna vaccine, where you're not giving the virus, but you're giving, the part of the virus that can, in your body, then make the proteins that look like the virus, and then that can stimulate an immune response. And then two other related newer platforms include DNA vaccines, and they're not really that new, but they're newer than a lot of the older vaccines. So instead of giving the kind of genetic instruction of RNA, you give a genetic instruction of DNA, or even giving the cells that would stimulate the immune system. So these are antigen presenting cells that have a little bit of virus bits on them on the top. That's another approach that's been used in vaccination. But I highlighted here just the two kind of categories that are being used in the context of SARS-CoV-2 infection. And this just shows it in a little bit more detail, where the viral vector vaccines, again, like the Johnson & Johnson, AstraZeneca, are really viruses that get into, here's a needle, get into the body and are capable of sort of, the immune system can take up those viruses and present them to the T cells and the B cells, so you can get an immune response in that way, just from kind of processing the viruses. And then these nucleic acid vaccines, so I mentioned the RNA vaccines and the DNA vaccines, which have the instructions for making the proteins that are injected, and then you have a cell in the body that has to make the viral proteins, and that's what stimulates your immune response. And the kind of really key thing for our pandemic response with the mRNA vaccines has been that they are fast to manufacture. So the viral vectors can be quick, but you can imagine that there's just a lot of quality. There's a lot of stuff that has to happen with making sure that the viral-based vaccines and the viral vector-based vaccines and even the protein vaccines takes a lot longer to make them than it does to make an mRNA vaccine. And that's one of the main reasons, I think, why we were in a good position to use these to quickly respond to the pandemic. So this is a really busy slide. It's not meant to kind of follow it all, and I've given reference here, and I'm happy to share these slides. But I just wanted to give a little bit of
Rachel, just a quick question to pause. What does mRNA stand for?
So nucleic acids are the building blocks of the cell. So in every cell in your body, do people know what DNA is?
A little bit.
Yeah. So DNA is kind of the code for telling every single cell what to do. So it's kind of the instruction book for every cell. The DNA has to then get turned into RNA and ribonucleic acid. And the specific kind of RNA that actually makes proteins that can go stimulate the immune system is called messenger ribonucleic acid, so mRNA. And so it's kind of like if you had like a really detailed instruction booklet, that's your DNA. But then you had, I don't know, we have a little baby, so I feel like we have a car seat that has like just the key instructions on it, like don't mess this part up. That's the mRNA, is just the little bit of like kind of telling you a specific thing of what to do. The DNA sort of gives you a sense of what to do overall in the cell, and the mRNAs say, in this case, can you make some spike protein, right? The spike is what we want to be able to make to stimulate the immune system. Yeah. Thank you, Lauren. So mRNA, the messenger RNA, was first discovered in 1961. And the main thing to know about mRNA is it's super, 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 super unstable. When we work with it in the lab, you joke that like you don't want to look at it the wrong way because it can just sort of break down and disintegrate. And so I would summarize a lot of what happened between 1961 and 2008 as trying to figure out how to like make this very unstable entity, how to kind of stabilize it so that when we give it as a vaccine, we know that it'll stick around and actually be able to do its thing. And a lot of key discoveries were made along the way, including a key one in 2008 to modify the RNAs to keep them stable. And I guess what I wanted to highlight here is that in 2009 was kind of people thought about using mRNA vaccines for a really, really long time because it's the instruction booklet, right? And if you can actually give the body the instruction booklet, that in some ways is the best way of thinking about like, here are my instructions, please go make an immune response against this virus. It seems like such a great idea, but took a lot of development. And in 2009 was the first time that the mRNA vaccines were used as a cancer treatment. And since 2009, there have been studies, and I think probably many people are familiar, prior to COVID, tens of thousands of people received mRNA vaccination, mostly in the context of cancer, but also in the context of other infections too, including some early and small HIV vaccine studies. The key thing for the mRNAs is that they need to be sort of packaged in something that makes them stable. And that got further and further refined over the years, such that when the pandemic kind of hit, this technology was really at a very mature state of understanding how to make it work well. And I think a lot of people appreciated all of the development that happened over these last 10, 11 years to make the mRNA vaccines really as potent as we have found that they can be in the context of COVID. So let me stop there and ask if there are any questions about up to that point in terms of that biology. Or ask you all what you think the challenges are for HIV vaccines.
What I heard, one of the challenges is that HIV goes into every pocket of the body, I guess is the way to say it. And there are pockets like your brain that can't take medication because it would damage those organs. So they're trying to get the HIV to come out of those hiding places to be able to kill them. And that's what's holding up the cure, is what I heard.
Yeah. So in terms of a therapeutic vaccine, the HIV virus can sort of hide in many parts of the body, and those can be challenging places for the immune response to get into. In terms of preventing HIV infection,
antibodies don't, you still have HIV when you have antibodies, right? So you could still have antibodies don't really neutralize HIV well enough, so that stimulating antibodies doesn't kill HIV.
Yeah.
So,
yeah, right. You can have a positive test for HIV, which is oftentimes an antibody test. So you know you have antibodies, but they're not working. And that can be due to many different things. And I think in my next few slides, I'll sort of get into that. But any other thoughts about sort of specific challenges for vaccine development, for like HIV, that, being able to work as a vaccine?
Alania just has a comment in the chat saying, I feel like I would participate in HIV vaccine trials and I would enjoy the ups and downs of trying.
Yeah.
It's not a challenge, but I'm curious if respiratory, if vaccines for respiratory illnesses are if there are different considerations for other types of conditions that's not respiratory in terms of making a vaccine.
Yep. So let me go ahead and share my screen. That's I promise we weren't collaborating in terms of what my next slide or my next concepts were going to be, Barbara, right? We weren't, you and I didn't chat ahead of time. Let me pull this up again. So there are a lot of different challenges, but in terms of thinking about developing a vaccine to prevent HIV infection, they're the kind of general considerations we think about for vaccines: safety, tolerability, scalability around the world, and of course, equitable distribution that need to be taken into account. But in terms of the kind of immunology or the biology sort of side of it, one of the main things we worry about with HIV vaccines is that we have a very tricky target here. So this is a picture of the HIV virus, and these things sticking out are a lot like the spike proteins that you hear about with SARS-CoV-2. These are called the envelope protein. And the two main things from the envelope protein are, number one, that I think many of you are aware, HIV mutates super, super easily, right? Are people generally kind of aware that HIV is, it has its instruction book, but when it goes to turn that instruction book into like making a new virus, it has like all sorts of errors. So almost every time that you get around, HIV is rude. I'm now seeing the chat. HIV is definitely rude. I like that. It introduces a bunch of errors sort of every time that it replicates. And what that means is if it introduces an error so that it can no longer be recognized by the immune system, suddenly that version of HIV is going to take over, right? Because it's like able to get away from those antibodies. It's able to get away from those T cells. And that, I think, is at the crux of like the hardest reason it is to make a vaccine against HIV. Is that this envelope protein is super, super easily mutated. So there are, tens of millions of different variants of HIV. We're here with COVID and we're like, oh, alpha and beta and delta and lambda and mu. HIV just sort of blows that out of the water in terms of how diverse the virus is, not just across the world, but within people. And that's right, Jesse, that's exactly why you get resistance to medication and why you need multiple pills to suppress HIV. So it evolves, and then I have here it hides, but I would say there are two ways that HIV hides. One is that like the HIV protein itself, I really like this picture. I don't actually think this is what it looks like under the microscope, but I just like it's kind of like wild hair, just kind of, so HIV sort of makes this it's sugar on the outside of it. And the sugar makes it really, really hard for antibodies to come in and actually bind to this envelope. So HIV virus hides, and then we were also saying that the HIV itself hides within the body really quickly. Within a couple days of infection, there's very extensive infection already within lots of the cells within your body. So we are talking about a vaccine having to work within 24 to 48 hours of getting the virus. And so there are a lot of challenges there. And then I think we were also sort of talking about the fact that we don't really know the best type of immune response against HIV. And I think one of the main takeaways is that it's actually different for every virus. So SARS-CoV-2, we make these great neutralizing antibodies from the mRNA vaccines. We have some T-cell responses too, but it seems really the neutralizing antibody responses are what's key to preventing this respiratory virus from causing, wreaking havoc in our bodies. Is that exactly the same thing we need for HIV? It doesn't seem to be the case, because we have had many HIV kind of what we call efficacy trials. These are trials that are designed to actually test, there are lots of early trials of like, can you make an immune response to this vaccine? But this is a list here of, and I think it's not fully exhaustive, but of most of the HIV vaccine trials. And they've really tried, if you remember the earlier slide, these kind of viral vectors like adenovirus and canary pox and DNA vaccination, and DNA and this thing called MVA vaccination. And with the exception of the RV144 trial that showed some protection, but then when the similar vaccine was used again in another trial, we didn't really see protection. We haven't seen that these vaccines have been able to actually stop the acquisition of HIV. And it's been super disappointing, I think, to see this. And I think the most recent trial that some of you may have read about is that a trial using the Ad26 vaccine. It's what the Johnson & Johnson, the J&J vaccine that was just like a one-dose vaccine for COVID. Same idea. And this trial was recently stopped because they were seeing that it didn't really prevent HIV infection. So there are a lot of different HIV, this is not, again, super overwhelming, don't pay too much attention to the left side other than to maybe get some hope that there's a lot of development going on in terms of approaching, making immune responses for HIV and developing HIV vaccines. But the key kind of, I think, newer ideas, or not newer ideas, but the things that we really have on our mind are that we need to be able to make immune responses that are going to be effective across these tens of millions of viral variants. And so are people generally familiar with the concept of broadly neutralizing antibodies or BNAbs? Sorry, that's my baby crying. Okay, so broadly neutralizing antibodies are essentially antibodies that can, it's exactly as they say, they can target wide range of HIV, even though it's mutated. So they kind of go after the parts of the HIV that don't really mutate. And they're neutralizing in that they can see the HIV virus and then neutralize it, sort of act immediately against it to stop it from going in and infecting another cell. So there's been a lot of improvement in the design of the envelope, the HIV envelope in the vaccine to enhance this. And I'll talk about how mRNA vaccination might actually help that. And then there's been a lot of work to elicit T-cell responses against conserved regions of HIV, again, that cannot escape recognition. So a lot of these designs are newer platform designs, also targeting this key concept that you really want to be able to, you don't have to worry about this in COVID. So SARS-CoV-2 doesn't really evolve in the body, but HIV does in response to the infection. And so prior to these past couple of years, most mRNA vaccine trials, like history-wise, were in a very different form from what we think about for the COVID vaccines. They were actually giving people immune cells with RNAs in them to try to stimulate the immune system, because we didn't have a great way to stabilize the mRNA in the body. And some of them were even given directly into lymph nodes. So these were kind of more exploratory, I would say, types of trials. But kind of to this question of whether vaccines themselves can, is it Alania? I don't know how to say your name. I'm so sorry. Am I saying?
I'm sorry, I had you muted, but my name is Alania.
Alania, sorry, Alania, you asked a great question. Is there a way to force the response?
Right.
So it's almost like, again, a planted question. So the latest thinking in HIV vaccine design is, unlike sort of SARS-CoV-2, where, yeah, you might get a few different doses, you can make your antibodies. Maybe we're going to have to eventually boost against, the Delta variant. But with HIV, the thought is that you'll really have to do sequential boosting. And now we understand a lot of the biology of how to make these broadly neutralizing antibodies, that maybe you're just going to have to do it again and again and again in a way to sequentially boost one after another and select for these broadly neutralizing antibodies that generally take a lot of time to make in the body. So, Eleni, if you want to come work in the lab and help, you totally have the right idea. So I just wanted to say that, these are all ideas kind of that are in the air about sort of, what are we doing with HIV vaccine design, and how can we take some of, what we know from other infections, and I guess in particular SARS-CoV-2 and COVID, and apply it to HIV. And I think that the biggest news in this field is that there are these kind of series of mRNA vaccine trials using the Moderna vaccine that were kind of the same thing that we're using for COVID. And using these vaccines, which again, remember, the RNA vaccines are, they can be made very quickly. You can kind of insert different sequences in them pretty easily. And it's easy to kind of think about giving multiple doses. And so the hypothesis here is that sequential vaccination can induce specific classes of B cells, the cells that make antibodies, and guide their ability. So it coax them into making, and I'm sorry, I wrote this as BNAbs. So these are broadly neutralizing antibodies. Yeah. And the two trials that you may have seen in the news, so one was, it's from Moderna and IAVI together, was a small study reported in February. I haven't seen the actual study, like I was trying to look for it so I could give you more details, but the kind of study by press release that I found showed that 97% of participants actually made these very difficult to make broadly neutralizing antibodies after being given this kind of vaccine regimen. And there's a kind of second trial that's going on that's enrolling now, and they're kind of slight variations that they're doing of exactly what's in the vaccine. But the idea is to be able to learn from each trial and improve upon what we're seeing to make better and better targeted antibody responses. So that's, I think, the biggest news in terms of sort of taking the technology, and obviously now a lot of safety information we have about mRNA vaccines, and applying it to prophylactic vaccines for HIV. And I would just, the last thing I had prepared was just like one quick slide to think about, how do we think about this for therapeutic vaccines? And whereas to prevent HIV infection, we want these antibodies that can neutralize the virus, and making BNAbs, broadly neutralizing antibodies, getting the B cell response is probably one of the main targets for a prophylactic vaccine. for a therapeutic vaccine, we really want to target these infected cells that are kind of persisting mostly in CD4 T cells, but throughout many, compartments within the body. And to do this, and this is really, again, to kind of come back to what my interests are, to do this, we really want to think about doing this with CD8 T cells, because CD8 T cells can find these cells and actually kill the cells that are infected with the virus. So how can mRNA vaccines help us there? It's not entirely clear. mRNA vaccines can induce a T cell response. But I think one of the exciting things about the mRNA vaccines, again, is the fact that they're a lot easier to produce and make if we want to think about sort of what do we want to target in the vaccine. And that may be very useful for thinking about in the context of HIV cure. Thank you, Lauren. Yeah, these are vaccines to help treat people living with HIV, as opposed to kind of what most of what I was talking about was thinking about sort of preventing HIV infection. So I'll stop there. I wrote down a couple of the things that we brought up at the beginning, and I can sort of revisit them. But do you have any questions? I know that was wasn't sure sort of how high level or detailed people wanted me to sort of get into it. And I'm glad we have a little bit of time for discussion to talk a little bit more just about, I think there are a lot of questions that come up in this context. And I think,
I have a question.
Yeah.
Elena here. I have a question. So when you were reading the one slide where you were talking about how it would have to be like continual treatments on a continuous basis and whatnot, does that mean it's something that would take years to eradicate? And if so, how would the body's organs respond to that? Like, would they shut down? Would they need treatment? Or would they be accepting to, said vaccine? Because I'm like Jesse said, if it hides in the brain and in crevices and cracks and all that, I'm a big girl, but I'll do it.
Yeah, so that's a great question. And I think the slide I was showing was mostly are you talking about the one about giving lots of vaccine doses over time?
Yes, that's what I'm talking about.
Yeah, so that's actually for making the antibodies for preventing HIV infection. So that would be to give to people to sort of prevent them from getting HIV. And it raises the kind of the question that, why can't we just take the vaccine for COVID and apply it to HIV? Mm-hmm. Well, it won't work as a single dose or even 2 doses for HIV. We probably would have to have this regimen to give multiple times to even prevent getting HIV. Now, for the studies where we want to try to cure HIV, people living with HIV and we want to try to get rid of it, mm-hmm. we probably wouldn't do this kind of broadly neutralizing antibody thing because people with HIV already have, like Greg said, we already have antibody responses against HIV. So it's actually, people are looking into this, but it's an even bigger challenge to try to figure out how to guide the immune response to make broadly neutralizing antibodies if you already have the infection. And so we're actually trying to do it for the T cells because we think the T cells are really important to get in and go find all the cells all in the body. But we have challenges with that. But there's a lot of interesting ideas and combining different things that kind of might be the right way forward. So sorry about my crying baby. Jesse, do you have your hand up?
So the $1 million question, how long do we have to wait for the cure? How close are we? 6 months, 10 years? And I'm angry that we have to wait to tell you the truth. Yeah.
I'm angry too. I want us to have a cure. I so want it. I have talked to every single one of my patients wants it in less time than wants it now. And it's not 6 months. I would say the mRNA vaccine development is sort of a sign of a lot of other development that's happening in terms of how we understand the immune system and how we understand to get the immune system to do what we want to do. And I think we are at a very pivotal time point for that, and being able to take advantage of, I didn't even mention this, but you probably all are aware of like cancer immunotherapies, to make the immune system fight cancer. Like a lot of people are being treated now to, do that. All of that work informs what we learn in HIV. And I think I don't have a precise answer for you. I wish I did. But I think we're getting a lot closer through all of that understanding, but I think it's going to require more of these clinical studies to really figure out how to get the immune system to do it. If anyone has a good way to talk to the immune system and tell it what to do, tell me, and we'll try to do it.
We're still at that answer. I've been getting that answer for years, so yeah.
I know.
Georgia, maybe we can take your
I don't
question.
know how to make.
Then we have to do the evaluation. So that
I'll
will
make
be
it
the last.
quick. It's a comment.
no,
I'm
okay.
Georgia.
Hi, Georgia.
I didn't know how to make my hand
Yeah.
go up any other way. I met Jesse when we worked in the Explore study years ago, which was a behavioral study. And I felt a rivalry with the vaccine team. Like, I always want to just solve all the problems with behavioral. Like, look, we know what to do, but people are complicated. So I love that you work with patients directly and you are working on the vaccine side. But I do find we gotta, somehow we have to work with both sides to keep people safe and keep people healthy. And I mostly in my job in HIV linkage in the Alameda County Public Health Department, the people I work with are the people with HIV as one of the least of their problems, but it makes it hard for them because of other challenges in their life to do the basic self-care. I mean, it's not all on them, it's the system. So I just wonder how you feel about balancing those things and that kind of rivalry.
Totally. We need every tool we need every tool in our toolbox, right? To get at this. I think HIV presents challenges. I talk a lot about the biology, but I see a lot of it in real life with my patients. We need every level of involvement and everything that we can do. It's actually so many parallels to COVID too, right? Like many of us, we know how to, prevent people getting COVID, but like we need to be able to do these things at multiple levels, including the biomedical interventions, as well as all the social stuff to really be able to support people and help them help avoid getting infected and taking care once you're infected and all of that. I see the survey. I'm regretful that I didn't have enough time to answer this question or really for us to talk about bridging the gap and kind of healing the wounds with pharmaceutical companies. They're not easy answers to that. I think it takes time, and I think it takes effort, and I think it takes willingness and openness to talk to people and have conversations and be willing to change course when, something doesn't fit right with the community, and making the effort to do educational work and things like that. But I would love to come back and have some conversations with you guys about just to talk more about that so that I can learn more than what I say. I think what you say is more important. So I'm regretful I didn't have enough time to discuss that. I got too dorky on the immunology side of things. Apologies.
Is there somewhere these slides are like available so I can look up words that I don't understand? Because again, I'm a patient myself and I really, really want to read that.
Yeah, I think Rachel said that she would make the slides available. I think I heard that.
Yeah, I can send them to Lauren.
Right.
Lauren, does that make sense? Or is there someone else I should write? Yeah, okay.
Yeah, and I just want to say thank you so very much, Rachel, and for the last statement that you would like to come back and continue to have the conversation with the CAB.
Yes,
I'm
definitely.
quite sure that they would love to have you come back. And so we'll be talking in the future. I thought your presentation was wonderful, and I can't speak for everybody else, but I was totally focused. So
Barbara,
Thank
are
you
we
so much.
staying here or are we going to a different Zoom? For
we're going to a different Zoom.
Okay.
So Rachel, we also have a CAB meeting right after this. We try to do the trainings right before our CAB meeting.
Got it.
And so that's, we're not rushing because we want to leave. We just have another meeting
makes
to go
sense.
to. But I
you.
really appreciate the conversation that you gave us, the presentation.
Yeah, thank you all so much for taking time to be here and to listen, and for being willing to talk to me again, hopefully in the future. And I think that would be really it'd be really good for me. I would really appreciate it. So
Thank you.
That's
all right.
great. Thank you.
Thank
Thank
you.
you.
Okay.
Have a good next meeting.
Thank you, everybody.
Thanks.